Hi there! Welcome to our video blog series. Come join us and our good friend Einstein to explore and learn more about what we do as synthetic biologists. We are the UCSF 2015 iGEM team, composed of 10 undergraduate students from all over the Bay Area. This is the second video in our video blog series for iGEM's Policy and Practices, a section meant to get our team to think of the bigger picture and what our project could do for the greater good of society. Our mission is to use the burgeoning field of social media to bridge the gap between the synthetic biology community and the general public. If you watched our video last week, we saw what the general public at a baseball game thought about synthetic biology and associated techniques such as gene cloning, genetic engineering, and some products of these techniques such as the creation of GMOs. While some liked what synthetic biology had to offer, many individuals were against it and raised some familiar ethical issues that surround the growing field. This week, we want to define these used terms in hopes to welcome the public into our world, increase knowledge about what we do in lab and the projects we work on, and erase the horrible, scary stigma of the mad, evil scientist that plagues the media around us. Let's start with the basics. DNA, the molecule of life, is the hereditary material present in cells of all humans and other living organisms. DNA can be seen as a code, a recipe, what makes an organism, or you. DNA is then transcribed into a single-stranded RNA. Finally, this gets turned into proteins that do most of the work in cells. Proteins are involved in basically every function you can think of. Gene cloning is often referred to as a way to cut and paste DNA. Genes are blocks of DNA that are instructions to make proteins. Many think of cloning of entire organisms like Dolly the sheep, but in reality, most cloning in science is the insertion of interesting and important single genes into other types of cells to study their function. With gene cloning, you have the ability to take naturally occurring or synthetic DNA pieces in which you're interested and recombine them to form new molecules. There has been a large effort to characterize, document, and standardize many of these DNA pieces we know about, which can be mixed and matched in cloning. We refer to these as biobricks. Genetic engineering is the process of introducing foreign DNA into an organism. Using some of the standardized DNA parts, biobricks, which we mentioned before, we can rationally design a new piece of DNA that consists of several of these biobricks that stack up like Legos, ultimately used to design or alter biological pathways in living cells. The rational design and construction of biological pathways and systems using these biobricks is the field we call synthetic biology. To design more systems, one can clone DNA from an organism or select desired DNA from the biobricks database, literally order it on the internet for pennies per base pair, and have it shipped to their door. Scientists then put together these bricks, typically into E. coli, a type of bacteria which is safe and easy to work with in a lab. We can use these bacteria to essentially photocopy our new DNA and make more, which can be put in an organism of choice. After the process of genetic engineering is done, a genetically modified organism, or GMO, is created. GMOs are able to make new products and perform new functions from existing organisms. They also come in all shapes and sizes, such as corn, genetically engineered to be herbicide resistant or produce pest repellent hormones. Or they can just come in a simple bacteria that can glow green. A lot of negative stigma surrounds the term GMO. In almost all individuals interviewed, people did not have a clear idea of what genetically modified organisms were, but media attention and controversy have placed GMOs in a negative light. Many believe that GMOs are unnatural and therefore have many risks associated with them, and that this might pose a threat to your health. However, GMOs are not always bad or scary things. GMOs have been used to make medicinal breakthroughs, new drought and disease-resistant food like frost-resistant strawberries, and GMOs have brought about revolutionary discoveries such as anti-malarial drugs and treatment for genetic disorders like Tay-Sachs disease, cystic fibrosis, and sickle cell anemia, which prevent oxygen from being properly transported around the body by red blood cells. It has also been used to do research on new biofuel sources. All in all, synthetic biology and genetic engineering have brought about many discoveries that have made life a lot easier. Through the tremendous potential of synthetic biology and genetic engineering, we continue to make groundbreaking discoveries that can largely benefit mankind. We hope that you learned a little bit more about what synthetic biology is truly about. Tune in the upcoming weeks for more videos on anything synthetic biology related, such as the bioethical issues that surround the field, as well as a closer look at our 2015 UCSF iGEM team members. See you next week!